Today's lecture is on cell walls, extracellular matrix, and cell interactions. So cells are usually surrounded by different types of secreted macromolecules, bacteria, algae, fungi, etc. These have rigid cell walls, but animal cells usually have some kind of extracellular matrix of proteins and polysaccharides. So the extracellular matrix is one that will help support the cells and tissues that play an important role in regulating cell behavior. And interactions between the cells are key to the organization and the communication of cells in the tissues of plants and animals. So cell walls determine shape and also prevent cells from swelling and bursting as a result of osmotic pressure. So bacterial cell walls are very different than those of eukaryotes. And bacterial cell walls will determine rod shapes such as E. coli, spiral such as pneumococcus, or spherical, such as pneumococcus, and spiral shapes such as Treponema pallidum, which is the cause of syphilis. Now, bacteria can be divided into two classes, gram-negative. They will have an outer and an inner membrane and a thin cell wall that's composed of peptidoglycan, whereas gram-positive have one plasma membrane but a very thick peptidoglycan layer on the outside. So if you take a look here, guys, here's the outer membrane of a gram-negative, the peptidoglycan here, and an inner membrane, and gram-positive has this thick peptidoglycan and the plasma membrane underneath. So the cell walls of eukaryotes are composed of polysaccharides. Chitin is the main one, and that also happens to form exoskeletons of arthropods as well. Algae will have plant cell walls of cellulose. And here's an example of the differences between chitin and cellulose down here. And you'll notice that there's different beta 1 to 4 linkages that take place in these molecules. Now, after there's transport that takes place across the plasma membrane, there's 36 cellulose chains that join in parallel to form what are known as microfibrils. The cellulose microfibrils can then extend for many micrometers in length. So plant cell walls are pretty interesting. They have these cellulose microfibrils, but they also have what are called hemicelluloses. Those are highly branched polysaccharides, hydrogen bonded to the surface of the cellulose microfibrils. They provide stability and mechanical strength. And here's an example of a hemicellulose molecule. Pectins are another important molecule. They are branched polysaccharides, and they have many negatively charged galacturonic acids that are cellulose, or that are associated. So they are cross-linked with microfibrils and basically bind positive ions such as calcium, and that traps water molecules to form gels. So gels, as you guys know, are pretty sticky. Now, cell walls in plants change as the cell develops. Our primary cell walls are relatively thin, flexible. They allow the cell to grow and expand. They have cellulose, hemicellulose, and pectins associated. Secondary cell walls are between the plasma membrane and the primary cell wall after growth has stopped. Now, they usually lack pectin and are about 50 to 80% cellulose. And they're further strengthened then by what's called lignin, and that's a complex molecule of phenolic residues. And again, if you think of things like wood, this is what provides the strength and density to wood. Now, animal cells are embedded within an extracellular matrix. A very important part of that would be basal lamina. There are thin layers on epithelial cells or where epithelial cells will rest. They also can surround muscle, adipose cells, and peripheral nerves. So here's your basal lamina up here, guys. Here's your epithelia here. They kind of stick right onto that. It's abundant in connective tissues. A lot of connective tissues have this, things like connective tissue, bone, tendon, cartilage, etc. And they're distributed throughout these different types of cells. So there's tough fibrous protein embedded in there, and we also have adhesion proteins that will link components of the matrix to one another and also to attached cells. So please know that the major structural protein is collagen, and it forms triple helices, and there's different domains, guys, that have repeats of amino acid sequence GlyXY, and glycine in every third position. We have type 1 collagen that will form collagen fibrils, and assembly of fibrils occurs outside of the cells from what's called a soluble precursor known as procollagens. Connective tissue with elastic fibers is common in organs, and as you can imagine, elastic means they stretch and return to shape. So think of the lungs. Your lungs have to expand and contract, so they have to be elastic. So elastic fibers are made of elastin. Now, extracellular matrix gels are formed from polysaccharides that we call GAGs, which are glycosaminoglycans, and they're repeated units of disaccharides. And here are the structures of those. Now, the addition of sulfate groups makes the GAGs highly negatively charged, so they can bind to positively charged ions and trap water molecules. And again, this allows them to form hydrated gels. So hyaluronan, I think I said it right, is the only GAG that is a single long polypeptide chain. All of the other GAGs are linked to proteins to form what we call proteoglycans, linked to form proteoglycans. So the proteoglycans will interact then with the hyaluronan and basically form large complexes. And then you can get an aggrecan, which is a major proteoglycan of cartilage. And again, there's about 100 chains of chondroitin sulfate that's attached to the core protein there. So this again, here's your chondroitin up here, guys, your aggregan, and then hyaluronan running through the middle right there. Okay, take a screenshot. Please take a screenshot and upload it to the Dropbox. So matrix adhesion proteins are very, very important components to stick cells to a surface and one another. Fibronectin is the main adhesion protein of connective tissue, and it's often cross-linked to fibrils. And here's an example of that. Here's your integrin, collagen, proteoglycan, and you've got these disulfide bonds that will hold these together. Now, our basal laminae will contain adhesion molecules of what's known as the laminin family. They have 3 polypeptide chains. They're rod-like. The subunits have binding sites for surface receptors, and they can actually self-assemble into networks. And here's a pretty cool example of that. You've got various chains on here. Here's where cell binding would take place, for instance, self-assembly at the ends of these 3 chains there. And they just keep sticking together, basically. So they're tightly associated with nitrogen, and that's an adhesion protein that binds to type 4 collagen. So you have a lot of networks that get formed, and that's the key thing to remember, guys, with a lot of matrix formation. Networks are formed. There's a lot of different components that stick together to keep everything in our bodies contained. Integrins, very important transmembrane protein that attaches cells to the extracellular matrix. These are really important, guys, because if you don't have integrins, what you find is if cells cannot attach to an extracellular matrix, they can sometimes undergo apoptosis, which is programmed cell death. Here's an example of matrix binding. And they were identified by immunofluorescence and different types of electron microscopy. They also anchor the cytoskeleton. So two types of cell matrix junctions. Please know we have what are known as focal adhesions. Those are bundles of actin filaments and are anchored to beta subunits of integrins by alpha-actinin, talin, and vinculin. So here's your talin here, guys, your vinculin. Here's your alpha-actinin, and you've got your actin filaments here. So it's helping the cell then adhere to the surface. The second type are called hemidesmosomes. They will anchor epithelial cells to basal lamina. They adhere them to basal lamina. And again, here's a blown up picture of that. You have a lot of different players in here, your integrins, BP180, pectin, etc., to allow that binding to take place. Focal adhesions can be very stable, or sometimes they will turn over very quickly, especially in cells that are moving along a matrix. So they have to be able to be flexible and if they're going to move. Now you can also have these clusters, or what we call focal complexes, that will take place, and they will be recruiting things like formin, and then that initiates actin bundles, talin, vinculin, etc., to all come together. And again, guys, here's an example of an inactive type of integrin. Now it's dimerized here. Then you get your focal complex, so you start attaching these together. Focal adhesions will take place over there. Now, animal cells modify their extracellular matrix with enzymes. They also will produce what are known as metalloproteases, which digest a variety of matrix proteins. And we talked about some of these in the one paper that we talked about with Alzheimer's and macular degeneration. So cell-to-cell adhesion is mediated by cell adhesion molecules. These will include selectins, integrins, different types of immunoglobulins, and cadherins. Now, the important thing, guys, is that a lot of these interactions with the adhesions require divalent cations such as calcium, magnesium, or manganese. Now, selectins will mediate what's known as transient interactions between leukocytes and endothelial cells, or platelets. And they can then bind to oligosaccharide ligands that are found on endothelial cells. Again, guys, endothelial cells are those that can be found in your blood vessels primarily. Okay, so here's your leukocyte, endothelial cell, that it will attach here, and then it can move along in that direction. Now, we also have these different interactions to integrins, and basically we have what's called heterophilic interactions, and that's basically when an adhesion molecule on one cell recognizes a different molecule on another cell. So you can have homophilic interactions, and that's when adhesion molecules on one binds to the same molecule on another cell. Cadherins will mediate these homophilic interactions, so please know that one, guys. And basically, guys, what happens is that they will stabilize adhesion junctions and link cytoskeletons of adjacent cells that are usually based on cadherins. Adherens junctions link actin filaments of adjacent cells. So again, these cells are right next to each other. And interestingly, we have a couple of different proteins involved in this, the catenins guys and P27. For whatever reason, they call them members of the armadillo protein family. I'm not exactly sure why, but maybe armadillos have more. Okay, and that's an example of some interactions there. And that's it for today.